Hello students, today we are going to study about anticoagulants. So they are EDTA, heparin, citrate, fluoride. So we will see them in detail.
So today we will see lecture on anticoagulant. So definition: anticoagulants are chemical substances which required to prevent clotting of the blood when it is mixed with the blood in proper proportion. So in short, we will see clotting mechanism. So when there is an injury to the vessel, so in first step the thromboplastin is released and then that is activated. Then in second step it activates prothrombin into thrombin, and then it activates the fibrinogen into fibrin monomer, and then all fibrin monomer combine to form fibrin polymer, in which all the WBCs, platelets get entangled, and then it forms the clot, and then it covers that injury area, and so that bleeding stops. Now we will see a few examples of anticoagulants. So first is ideal anticoagulant used in hematology. It is EDTA, ethylene diamine tetraacetic acid. So the mechanism mainly it is calcium chelating agent. So it removes calcium, and so that there is no clotting, and then blood is in liquid state. So uses of is all hematological investigations like HB, CBC, ESR, PCV, and all cell counts. Second is trisodium citrate. It is 3.8 to 3.2%. Mechanism mainly it converts ionized calcium into unionized, and because of that then they are not available for the blood clotting. Uses mainly by ESR by Vesegrin method and prothrombin time, or blood coagulation tests like PT, PTT, APTT, etc. Third is sodium fluoride. So mechanism mainly it is prevent glycolysis of the sugar in the blood, and because of that the blood sugar level is maintained, because it prevents action of enzymes, glucose oxidize, and uses is blood sugar level. Other anticoagulants: first is heparin. Action is antithrombic and use mainly it is for the blood gas analysis. Next is blood that is double oxalate. It is ammonium oxalate plus potassium oxalate, and then uses are like EDTA. The potassium oxalate it is mainly used for the blood urea level. Plain bulb, where the no anticoagulant is added, and it is mainly required for the serum preparation, which is required for the biochemical and serological investigations. Anticoagulants used in blood bank. So first it was ACD, that is acid citrate dextrose. Then it was CPD, that is citrate phosphate dextrose, and now it is used is CPDA, that is citrate phosphate dextrose adenine, or sometimes SAGM, that is saline adenine glucose mannitol. So we will see functions of each component. So citrate it acts as a anticoagulant, phosphate it acts as a buffer, dextrose it gives nourishment to the RBCs to be kept for more days, and mannitol it is a RBC membrane stabilizing agent. So ACD blood can be stored up to 21 days, CPD blood can be stored up to 30 days, CPDA blood can be stored up to 35 days, and SAGM blood can be stored up to 42 days, and this SAGM mainly used in a high setup blood banks. Hello students, today we will talk liquid portion separated from blood after clotting. So in that anticoagulant is not added, and because of that clotting mechanism takes place. Plasma is liquid portion separated without anticoagulant, and therefore clotting mechanism doesn't take place. So clotting factors are not present in serum, but clotting factors are present in plasma. Serum is used for preparation mainly for the investigations of biochemistry or serology, and then in that plain bulb you have to use for collection of the blood. Plasma is used mainly for the blood coagulation tests like PT, PTT, APTT, or blood sugar test like fluoride bulb is used, and for blood coagulation test citrate bulb is used. Thank you.
These are my references from which we can study. I hope this session you will find useful. Thank you.